Well, welcome to your lesson on carbohydrates. We're going to talk about these macromolecules in this particular unit. There are 4 types that we care about. Carbs is the first one. If you look down here, we've got our list of the other 3. So carbs we'll start off with, then we'll talk about proteins and how we put those together. We'll talk about lipids, which is the fancy word for fats. And then we'll also talk about nucleic acids as well. So organic compounds are really any molecule that contains a heck of a lot of carbon and a whole lot of hydrogen. Inorganic compounds can be anything else. The main difference between them, because these, like you can see carbon dioxide here, obviously has carbon in it. You can have carbons with hydrogen that are not organic. But so then the basic definition that you can do between the two is that, did you need a living thing to make it? Or is it commonly created by a living thing, or is it just, it can exist without living things? So water, oxygen, carbon dioxide would be inorganic because although we use them as living organisms, they can exist without life. So we can find these gases floating around in space somewhere or water floating around in space somewhere, and we don't say, oh, look, there's a sign of life. But if we start to see things that are built sort of like a sugar or like a protein or something, we can say, hey, look, there's life there, because that's a type of molecule that tends to be associated with living things. So organic versus inorganic, that's the basic idea. So again, the macromolecules then, what we're saying is we're looking at the giant large molecules. We're not looking down at the small little waters and oxygens and carbon dioxides at this point. We're looking at these huge monsters of things, just like you saw in that vitamin B, where there was just carbons all over the place. It was a really huge molecule. We're looking at the same thing here, really, really big molecules. All 4 of these types of molecules are going to be joined in the same way and broken up in the same way. You're going to join them by removing a water, and you're going to break them apart by adding a water into that gap. So we'll go through that. It'll be consistent between all of them. And with the exception of your fats, which you'll see in the next lesson, these 3, carbs, proteins, and nucleic acids, are basically simple subunits that you then click together to make longer and longer chains. Sometimes those chains are able to branch off in different directions. The carbohydrates are able to do that, and you'll see when we look at the structure why that is. And then other things like the proteins and the nucleic acids, they don't branch off. Imagine that there is no branches and it's just these 5 dots in a row. They're just click, 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 click together rather than clicking off at different directions. So we would say that they are, in this picture here, you're seeing a branched chain. And if you didn't have those branches, you just had the 5 in a row and nothing sticking off, then you would say it's an unbranched chain, okay? So you're just clicking these common things that are the same every time, and you just click them in together, and next thing you know, you got yourself from a monomer, mono meaning one, to a polymer, which means more than one, many. Poly means many. This will be a key thing after we talk about lipids and on your first quiz and your test, to say that lipids do not form polymer chains. There's a freebie answer there for one of the multiple choices. So let's get then into the actual carbohydrates themselves. Carbohydrates, what they are is built right into the name. You have carbon, and then hydra is the water part. So it's not water specifically, but what it is a ratio of the number of hydrogens to the number of oxygens is always 2 to 1. So they always have carbon, hydrogen, oxygen, and the H and the O are always in a 2 to 1 ratio every single time. That's how you can tell the difference between a carbohydrate, which is CHs and Os, and a fat, which is also CHs and Os, but there's no 2 to 1. So carbohydrate, 2 to 1 ratio. Make sure that you write the C's over. I know in your diagram it's got the short form, but it is important when you go to draw these that you have all the carbons drawn in. So if you could add those in, that would be great. Here we're getting into the most simple type of a carbohydrate. You're going to see that we're going to have these single sugars, which are these guys here. Then we'll be able to stick them together as double sugars. And then from there, you can just keep adding on these sugars, and you end up with these huge, big, long chains. Okay. So monosaccharides then are the simplest kind. I need you to remember that the 3 kinds that we care about for our course is glucose, fructose, and galactose. What you're going to notice, if you were to actually count up all the carbons, all the oxygens, all the hydrogens, you would see that they have the exact same molecular formula, so what the actual molecule is composed of, but you can see at a glance that the structural formula, so what it actually looks like, the way it's laid out, is very different, especially between glucose and fructose. And having these laid out this way, even though they have the exact same component parts, having them laid out differently like that makes them structural isomers. So the same molecular formula, but you have a different location for those parts, so you have a different structural formula. And then you can give me these exact things. You say, oh, like any monosaccharide, like the glucose and galactose, glucose and fructose, or so on and so forth. So that's another question on your test there. So here what we have a 6-carbon sugar, but we have 1, 2, 3, 4, 5 carbons that are actually in the ring, and then 1 that's popped out. And then with the fructose, we have 1, 2, 3, 4 in the ring. 1's popped out here, and this one is popped out over here. This will come back into play, the difference between these two shapes, when we talk about glycolysis in the cellular respiration unit, so in our next unit over. That's why I want you to kind of have at least an understanding of glucose versus fructose. But what I really want you to do to be tested in this unit is to memorize this structure right here. Now, it's going to be helpful if you understand that this is a 3D molecule. We draw it in 2D because how else can we do that on a 2D piece of paper? But it is a 3D molecule. So what I want you to imagine now is that you're out in the backyard and you're looking at the trampoline. Okay? You have the trampoline surface, that's like the ring that you see here, and then you have legs going down. And for those of you whose parents don't trust you and think you're bad at jumping, you also have the net that goes up, right? Can you picture that? So now when you're looking at this, I want you to see it in 3D. I want you to see this blue ring here as being your trampoline. And then I want you to see there's a leg going down, leg down, leg down, leg down, leg down. Can you see the legs? And can you now see the net going up? That's up, up, up. This H is up and this OH is up. Can you see it? And picture it. So now you've got the 3D version in your mind, okay? And now I want you to memorize this. Down, up, down, down. What am I talking about? I'm talking about this little functional group right here. Do you remember the name of the functional group? That's OH? Hydroxyl group. Okay, so now we have a hydroxyl pointed down. Then we have one up. We have one down and one down. You see that? That's what I mean when I say down, up, down, down. Down, up, down, down. When you come and you look at galactose, is it down, up, down, down for galactose? You have up, up, down, down, right? So if you take a look here, this one leg, instead of being a leg like it is here with the hydroxyl, in the galactose molecule, it's actually sticking up instead. So even though every other portion between the glucose and the galactose is identical, the fact that the hydroxyl group is in a different orientation, up versus down, means that our body does different stuff with this molecule. This is why functional groups are so important. It not only defines the characteristics of how it will interact with another version of a molecule, but it even defines how our bodies can use this sucker. Because having that hydroxyl up versus down means that I've physically got a different, like I've got a heavier, lumpy thing up and a little tiny hydrogen pointed down. So when I come with my machines to try and break this apart, there's going to be hydroxyl in the way for the enzyme that's supposed to come surround the glucose and rip it apart. It can't get in there because there's something in the way, physically in the way, okay? It would be like trying to shut the door, but somebody's got their foot in the way. If there's a physically something there that doesn't allow you to use the machines that you would normally use on glucose. So galactose and glucose, even with that tiny little switch, are going to be used differently in the body. Where the functional groups are, even their orientation, still attached to the same carbon, even their orientation matters, okay? And then obviously fructose is a lot different than either of these guys, so I'm really going to have a hard time trying to interchange glucose and fructose. My body's going to use it differently. So just keep that in mind. You have to memorize the structure of glucose for sure. You don't need to memorize or draw the fructose for the test, and you don't need to memorize and draw the glucose, or the galactose, pardon me, for the test, but you do just need to know the difference there. So can you spot the difference? Absolutely. We have the reversed orientation of that hydroxyl. So these are your 3 monosaccharides, right? Mono, because they are a single sugar. And then the word saccharide actually means sweet. So it's a single sweet thingy is actually what the name means. Glucose, the word gluc also means sweet. So when we're describing the sugar, we're saying it's the sweet version of sugar. Not all sugars are as sweet as other types of sugar. Fructose would be literally your fruit sugar. So now we're going to get into our disaccharides, which is when we take 2 of those smaller sugars and we shove them together. But we have a very specific way that that's going to happen. And it's through a process called dehydration synthesis. Dehydration synthesis. the name means exactly what it's going to do. If you're dehydrated, what have you lost? You've lost water. So if you are dehydrated, and that means you've lost water, what does the word synthesis mean? Create, to join things together, to create. So what I'm going to do is I'm going to dehydrate, so I'm going to remove a water for the purpose of joining some molecules. And you're going to see dehydration synthesis is going to build carbs, proteins, fats, and nucleic acids. Every single time we do the exact same thing, we remove a water, and that leaves us with open bonds that we can then pair together and click these little individual molecules together. So then we've got, in this case, what we're going to use is the two hydroxyl groups that are kind of sticking off the side here. And what we'll do is the O will stay behind on the glucose, but the hydrogen of that hydroxyl group is going to get ripped off along with the entire hydroxyl group here on this sugar. Now I could do glucose and fructose, I could do glucose and glucose. Any of these sugars you can shove together. We're just using the visual example as glucose and fructose in this case. Once I pull those guys off, if you can imagine then, this oxygen would be left behind. How many bonds can an oxygen make? It can make 2 because it's got 6 electrons in the outside shell, so it just needs 2 more to pop in, right? So here we haven't ripped this bond off, it's still joined to this carbon here. So that's still a bond there, but once I rip this hydrogen off the oxygen, I'm going to have a spare bond available. Same thing over here, I have a carbon sitting here, and once I rip this hydroxyl off, this carbon, which has 1, 2, 3 bonds already, this 4th purple bond that I've drawn here, it will have to be filled in as well. So what do you know? Once you remove that water, you're going to get that oxygen will then be paired up with this carbon on the right. And that's exactly what's drawn over here. That original oxygen is paired up with the original carbon. But I have removed 2 H's and an O, and when they get joined together, obviously 2 H's and an O make dihydrogen monoxide, water. When these guys get joined together, specifically glucose and fructose, this is where you get your table sugar from. This is another one of those facts that you're going to want to memorize. It's one of those simple little, quick little marks that you're going to want to be able to recognize in a multiple choice. Glucose plus fructose equals sucrose. When we do our drawing test, I'm going to want you to do glucose drawn next to another glucose, draw that, and then I'm going to want you to produce maltose, which is the double sugar that only has glucose in it. If you've ever had a malt beverage, then that's what this is. This is malt sugar here. So sucrose for your normal table sugar that you throw in your coffee or whatever. And then if you've ever added anything malt, like a malt root beer or something, it's this type of sugar that's causing that flavoring. It's got a slightly different taste than just regular sucrose. So in addition to understanding how they're kind of joined together, where you're ripping out the water, so you're dehydrating it for the purpose of synthesizing them together, okay? There is another word that we use to describe that particular reaction, and that's this condensation word. You already know what condensing is. You had your elementary school and grade 9 experience of learning the water cycle. And so you have water evaporate, and then when it cools, it condenses. Really, condensing is the idea that water droplets are forming, or water forms and seems to form out of nothing. It's clearly not. It's out of water vapor, but it's that the water is suddenly forming and falling from the sky. So we're using it in the same context here. Water is being formed, so we call it condensing. You can either use dehydration synthesis, or you can use the word condensing or condensation when you're describing things. You do need to recognize that these are the exact same thing when I'm describing things. Just so going forward, you know that they're interchangeable terms. Before, when you took a look at the formulas, let's take a look at the original. All the monosaccharides were C6H12O6, every single one of them. Please memorize that if you haven't memorized glucose already. You might have already memorized it in other courses. If I'm going to take a C6H12O6 molecule and then put it next to another C6H12O6 molecule, but I'm going to remove a water, what atoms will actually be joined together? How many of each? We're going to end up with one monosaccharide plus another monosaccharide, but removing the water. When you add the carbons together, 6 and 6 is 12, okay? 12 and 12 is 24, but I take off 2 of them, so now I've got 22. And then, of course, 6 and 6, but take away the 1 oxygen, I'm going to end up with 11 oxygens. So this is another easy one to memorize. C12, H22, O11. If you forget it, you can always just because you've memorized the monosaccharide one, you've memorized glucose's formula, you can always just do this, add them together and subtract the 1, as long as you remember to take out the water. One of the questions on the quiz could be, what's the formula for a trisaccharide? Well, then you just have to think, well, I'm putting 3 of them together, so it's C18, H36, O18, but in that case, I'm going to remove 1 water between joining the 2, and then when I add the 3rd one in, I'm going to have to remove another water. So I just take off the 2 waters. So it's C18, the 36 minus 2 and 2, so it ends up being 32. And then the 18 minus the 2 oxygens becomes 16. So that might be the type of question that you'll see. What I want you to notice, though, when you're looking at the formulas, C6H12O6, the original monosaccharide, H12 and O6 is a 2-to-1 ratio, right? Here again, we're seeing 22 to 11. Well, that's a 2-to-1 ratio. And even in the trisaccharide, 32 to 16, that's a 2-to-1 ratio. This is why we can define this as a carbohydrate, because of the 2-to-1. And again, that'll be different when we get to fats. The monosaccharides, just because of kind of like their nature of, they're quite small. The carbons compared to the number of hydrogens and oxygens is much smaller kind of a ratio kind of a thing. So what you'll actually find is because of this, there's more oxygen compared to the number of carbons. The fact that the oxygen is an electron hog in covalent relationships is going to matter when they're small. It matters when they're small. So that you'll actually end up with a carbohydrate if they're the small ones, the monosaccharides, and even in most cases the disaccharides, they're small enough that you actually get this polar relationship happening. So you'll get a slightly positive area on the molecule and a slightly negative area on the molecule. Why does that matter? Because water is polar, and because water is polar, it interacts with other things that are polar. So let me just go back to that note we had earlier. So here's where we talked about the polar nature of water. And you can see here that you've got slightly negative end where the oxygen is, slightly positive where that is. This means that if I throw in something else that's slightly polar, like a sugar, like those tiny sugar molecules, they're actually going to be able to come in and sit in between the water molecules, and they will lock into place as well. And they will fill up all these different gaps that we have here. You'll constantly have the sugar coming in. This is why sugar dissolves in water. It's also why salt will dissolve in water, because they can fill in the spaces between the liquid water molecules. Okay? And they do it very well. If they are much larger sugar molecules, so I don't just have a monosaccharide, one single sugar, and I don't just have a double disaccharide sugar, but I have much longer chains of them, then overall they're no longer polar, and they no longer interact with the positives and negatives here, and so they won't dissolve in water. So we have this fancy thing that we say, like dissolves like. Okay, and what do we mean when we say that? We can say that polar or polar solute dissolves in polar molecules, or the solvent. The reverse is true as well. Nonpolar dissolves in nonpolar. So now you're thinking about, well, what about oil versus water? What's the deal there? Well, shortly you're going to learn that things like oil would be considered nonpolar. Water is considered polar. So will oil dissolve in water? No, and you've already experienced that. So oil and water are not going to mix. When we get to cell transport, this is going to explain why it is that some particles are able to freely cross the cell membrane, because the cell membrane is made of oil. It's made of a lipid. And so anything that is oil-based is able to just freely come through the cell membrane, because the cell membrane will allow it to dissolve and then it will pass it to the inside. But if it's a water-based particle like a protein, it's not going to be able to come in without having a trigger or a special channel to get it through that oil. So oil and water aren't going to mix. So we would say then that oil, which is non-polar, because it doesn't mix with water, we would call it hydrophobic. So anything that's non-polar, hydrophobic. Anything that is polar is what? Hydrophilic. Friendly to water, scared of water. This is kind of how all these things are going to begin to kind of come together with our new terminology. So I really want to try to wrap your head around it and commit these new terms to memory, because you're going to want to start using them. The best way to learn a new language, I always say this, best way to learn a new language when you're in French class or whatever is to actually use it. That's why the French curriculum has changed in the last few years. So the words that you use are the words you're going to keep. And that's the same thing that goes for anything in English. Any new terminology. If you begin to use hydrophobic, hydrophilic in actual context, you'll remember it. And you won't have to be like, shoot, what's that one again? Or oxidation reduction, those types of things. As long as you start to begin to use those words in their context, you're going to be fine. So when I suggest to you guys that you have your neighbor explain it to you and you explain it back to them, please take advantage of those opportunities, because they are meant to lock the memory into your brain. You can say it out loud, you can hear it, you can see the words spelled so that you have that visual memory as well. And it really does help you to get through this first unit. Plus, again, all this terminology gets used for the next couple months, so you want to make sure that you're good at it. So the monosaccharides and the disaccharides then, because they're so small and you have more of an influence of the oxygen pulling electrons away from these carbons in the ring, you're going to most likely have a polar molecule, which means it can dissolve in polar water. It's hydrophilic. But as you get longer and longer, so imagine that these little dots here are different glucose. As I add glucose to glucose and maybe I even branch off, I don't have to always use this hydroxyl. Can you imagine if I used this hydroxyl, I'd head off into this direction. I'd go down. So that's where I'm going to get my ability to have branch chains with these sugars. So let's say I just kept going and going and going. Now overall, there are so many carbons and hydrogens compared to the number of oxygens that overall that ever so slightly positive and negative areas completely cancel out, and I get myself overall a non-polar molecule. So these big suckers are non-polar. If it's non-polar, it means it will not dissolve in water. So you can have simple sugars, either mono or disaccharides. Simple sugars are soluble. Complex carbohydrates, like these suckers, are not soluble in water, which is good because when you start to see what these starches are, you'll appreciate the fact that they are non-soluble. So polysaccharides now, poly meaning many sweet sugars, or what we call our complex carbohydrates, and these are the long chains of monosaccharides. We have 4 terms in general that we use that I would like you to memorize. If we're talking about the stored energy version, in plants we call that molecule a starch, and in animals we call that molecule glycogen. Very similar idea, that they're big long chains and they can be branched. The main difference between how a plant stores its sugar and how animals store their sugar like us is how branched they are. So this one's every 10 to 12 subunits, so you'd have 10 or 12 of these before you have a branch. And here there's more branches in a glycogen, so every 8 to 10. So that's the main structural difference. I don't really need you to memorize that. What I need you to memorize is that it's stored energy. We call it starch in plants. We call it glycogen in animals. That's the main thing. Where would you store it in plants? Well, this is all your white parts of your plants. So if you think about your potatoes and certain chunks of the stems and things like that, usually roots and tubers like potato stores the starch in that white section. Rice would have a lot of starch in it, so on and so forth. If it's an animal, we have glycogen mostly stored in the liver. White blood cells carry some glycogen themselves. And then in the muscles. And the reason for that is we want to have quick access to glucose when we need it. Glucose is going to be the thing that powers our actual cells to be able to do any and every job that they want to do. If they want to move something in and out of the cell membrane, they want to eat or they want to get rid of waste, they use glucose to create the energy for that. If you want to be able to flex a muscle, anytime you do that, you're burning through sugar. That's why you're going to have your temperature heat up as you begin to go through cellular respiration, because heat is a byproduct of burning sugar. Burning it chemically. We're not burning it like a lit match. So glycogen then mostly stored in the liver there, but also close to muscles, because muscles have a huge energy requirement. They got a lot of mitochondria burning through a lot of sugar. And if I can store it close by, then all I have to do is every time I need a glucose, I just chop off the end of this and now I've got a free glucose that I can use. So you tack them on by dehydration synthesis and you cut them off in order to use them later. When you are thinking back to grade 11, we talked about the digestive system. We did the reverse of all of these. We said, okay, you start off with your big complex carb, your big amylose, and then you cut it down to maltose, and then you get to your glucose, and you're chopping things down. In this case here, we're talking about now you're building things up. But when you do eat complex carbs, it does take a fair bit of work for them to... You can't just take this huge big carb and then suck it into a cell and throw it into the bloodstream. You have to chop it down first, and that's where your digestive enzymes are going to come in. I think you hopefully remember in grade 11 you talked about your spit has amylase in it. That's taking the big sugars and chopping them down into double sugars, into maltose. Then it goes through the stomach, and the amylase enzyme itself gets damaged by the stomach acid, so it breaks. So then on the other side of the stomach, in the duodenum, that first part of your small intestine, you've got more amylase being squirted in from the pancreas. Do you remember that, pancreatic amylase? And when that gets squirted in, it continues the process of chopping these huge, big sugars down into single, double sugars. Now that they're small, now I can suck them into the bloodstream and go and process them somewhere. Poly are too large to be absorbed by cells in the digestive system, have to be broken down. They are insoluble in water. In plants, when we're talking about the structure of a plant, we call it cellulose. It's very similar to glycogen with one major difference, and I'll show you what that difference is another day. But cellulose is a huge big polysaccharide that is in the actual plant cell walls. A lot of the clothing that you're wearing right now, if it's not a synthetic fiber, you're probably wearing a heck of a lot of cotton, right? Your jeans, your t-shirt material, all that kind of stuff, that's usually made of cotton or some kind of a cotton blend. Well, cotton comes from the cotton plant, and specifically, it's sugar. It's the cellulose sugar that they refine out of the cotton plant that they then, those fibers are what become your clothing. Same thing with the paper that's in front of you. It's the cellulose from the trees that forms that paper fiber. Okay. So in that case then, please appreciate that when you go out into the rain, your polystarches are not going to be soluble, so that when water hits you, your clothes don't just dissolve on you. So plants, structurally, we have cellulose. It's not soluble because it's so large. When you're eating cellulose, eating plants, you actually cannot digest it. You have no way to do that because you don't have the enzyme tools to help break them down. And so because of that, we call this fiber when it's in the human diet. It goes through you completely undigested, but it's still incredibly important in your diet because it's going to help to clean your colon, and it's going to help to attract all this water and this mucus and trying to keep things moving through the digestive system. So we call that bulk or fiber in your diet. Very, very healthy for you, because as long as you're constantly cleaning out the colon, you're not going to be worried about leaving junk behind. You're not going to leaving junk behind in the colon and having a very unclean colon can actually lead to all sorts of different problems, even things like colon cancer and stuff like that. It can cause disease in the colon. So you want to make sure you got a nice high fiber diet, keep things moving so that you don't end up with some kind of problem. So in plants, structurally, we say cellulose. In animals, we call it chitin. Again, the CH makes a K sound, so chitin. And this is very similar to cellulose, but it's just the animal version. Just like when you looked back up here and you saw between starch and glycogen, they're basically the same, with the exception of how often you have the subunits. It's the same kind of a thing down here. Very similar, good enough just to know that they're similar. The point being, what you want to remember is it's a structural version of sugar. In plants, it's called cellulose. In animals, it's called chitin. Okay. Where do we find chitin? Well, the last time you squished a bug, I'm sure it was by accident. The last time you squished a bug and you heard that crunching sound because of their exoskeleton, that's made of chitin. You've got fingernails, things like that, made of chitin, okay? This other type of a sugar. We've used chitin to our advantage. If you wear contact lenses, you can have the disposable kind, or you can have the hard glass version or plastic or glass version. The disposable ones are usually made of sugar. They're usually made of this chitin stuff. But it's insoluble sugar, so that when you pop it on your eye, it doesn't just dissolve on you. So we can use it for all sorts of little weird things like that. Including, anyone ever had stitches that you never had to pull out again? Usually they do them in the mouth. Anybody had their wisdom teeth out yet? But if you have any kind of stitches where they don't go back in and try to take out the stitches for you, it's probably made of a dissolving stitch, which is made of sugar. Something like a chitin sugar. Just grab an atom and just, if you're clicked on C, then you can just put a C in. Okay? So you can build your little molecules. In this case here, what I'm going to do is build sugar. I'm going to build a glucose specifically. Okay? So take a look on your page, look at the glucose picture. So what have I got? I've got a ring of 6, but one of those things is an oxygen. So I've got 1, 2, 3, 4, 5 carbons in the ring, plus an oxygen. Okay. Then I've got a carbon that goes up, right? And that's surrounded by an OH group, plus a couple hydrogens. Right? And then I've got all my down, up, down, ups. Now I can just go over here. I can grab myself a single bond icon, and I can join those guys together. Because it's for organic chemistry, it can anticipate that I already need a C because there'd be so many carbons, but we're just not going to use them. I'm going to draw it like that. Okay. And now I have to consider all the hydrogens and the oxygens that are coming off of these guys. So up here, I'm going to have an O and then an H, so I have an OH group. And then this carbon is going to only be joined to this carbon and this oxygen once, and then I'm going to have another hydrogen up here and another hydrogen up here. That's why it's written out. If you see there, it says it's written out as CH2OH. That's because that's just the truncated form of it, just to make life easier. So when you're drawing it out for marks, you can either write the CH2OH, or if it helps you, you can draw all the bonds. For this particular course, I'll accept either. So there's the CH2OH up there. And then what I'm going to do is, from memory, I'm going to do the down, up, down, down. What did I mean when I said that before? Hydroxyls, okay? So I'm going to just remember that. I'm going to say it's down, up, down, down. And then I fill in the hydrogens. There'll be hydrogen there, hydrogen with that guy, hydrogen with that one. Quickly do those bonds. And of course, you'll be drawing this out yourself. So you don't have to use MolView to do this. You can just do it on a piece of paper. Okay. And then I have to fill in the rest of the carbons with just H's, because every carbon should have 4 bonds. So I have 1, 2, 3. That tells me there's something missing here. In this particular one, because I've taken care of all the important things, the CH2OH is done, and all the hydroxyls are in place, I know that the rest of the things I have to fill in are hydrogen. So 1, 2, 3, 4. And the one that people always forget is the one that belongs to this carbon right here. So there should be a little guy right there. So then you just join them in. Just take a second here to do that. I'm just going to drag this down a little bit, get it out of our way. Uh-oh. I have to move that away first. Otherwise, it'll think I'm replacing it. I just want this bond to show. Okay, so now I've got a complete molecule. You'll draw it a little better than what I've just done here, where it actually looks more like a hexagon, please, than what I've done here with the computer. It's a little easier when you're actually doing it by hand. But that's the basic idea. You're going to be learning to draw these molecules. So the very next couple of pages in your booklet are going to give you a chance to do that, to draw things out, pay attention to where things are, and all that kind of stuff. What I want you to make sure that you always do, though, let me just switch screens. So here I've got my carbons, and you see I've laid the top and the bottom carbon are kind of on top of each other. This helps me to make a nice, consistent hexagonal shape to it. And then I just join those guys because they're all going to be single bonds, so that's nice and easy. And then I go and I do my CH2OH above. CH2OH. So that's done. Notice that I've done something very specific here, though. I've joined the carbon in the ring. In real life is joined to the carbon above, right? So I have to show that join. I can't put it into no man's land. If you draw me something like this, let's say that this is that same carbon, but you just went like this, and then you kind of shoved that bond up into no man's land, it's wrong. You can't just shove the bond into no man's land. You have to show me the bond is joined, which 2 atoms are actually connected. And it is the carbon that's actually connected there. And again, if you were to draw out that little section, it would be, here's that one carbon. It would be to a carbon that's attached to the OH group, that's attached to a hydrogen, that's attached to a hydrogen. So you're trying to simulate that same bond, even though you're doing the short form for it, okay? Same thing goes for all of your hydroxyls and your hydrogens. You have to make sure that it's the oxygen connected to the carbon. So you would have to draw it out like, here's down, up, down, down. And notice at no point in time, I never did carbon sort of attached to a no man's land. That's wrong. It has to be attached directly to that oxygen. So you have to be very clear that this is who is being joined here. Now notice as well, I've done HO or OH. It doesn't matter. Usually you'll have your hydrogen sticking out of the molecule. So just whatever's convenient. I'm not going to mark you right or wrong on whether it's facing in or facing out. I don't care. So as long as the bond itself is correct, you've got the O attached to the C, then you're golden. So far I've done the ring. I did the CH2OH next to get that off of my memory list. And I've done the down, up, down, down for those hydroxyls. Everything else is a hydrogen, so it's not that hard to remember once you get going on it. Hydrogen. Don't forget this little pesky hydrogen that comes off that top C. Hydrogen, hydrogen, hydrogen. And if you want to make sure you haven't missed anything, count up the bonds. So you should have, for every carbon, you should have 4 bonds. 1, 2, 3, 4. This one has 4 bonds because it's 1 down, 2 going to that H, just like it is over here. So you have 1 down, 2 going to the 2 H's, and then 1 going out. All right? Then you have this carbon, 1, 2, 3, 4. Carbon, 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4. This is how you don't miss little pesky hydrogens. And I call them pesky because people lose half marks all the time. And they're like, oh, I knew there was a hydrogen there, and they just didn't count their bonds. And then, of course, your bonds have to be joining the right molecules. The carbon does not join to the hydrogen. The carbon has to join to the oxygen. And then having the hydroxyls in the right spot, all that kind of stuff. That'll get you all your marks. Okay. Memorize glucose. You do still want to draw galactose and fructose at least once because you're counting up the number of carbons, hydrogens, and oxygens, and you're proving to yourself that yes, there is actually the exact same molecular formula, even though the structures are different. When it comes to building the molecule kits, if you're in class, I can grab the actual balls and sticks for you if you want, or you're welcome to try that MolView program if you want, either way. The nice thing about the MolView program is that it built the 3D structure over on the right-hand side for me. So then I could grab it and I could flip it around and I could manipulate it and take a look and see what it's supposed to look like with the bond angles. Okay? If you find on this assignment that some of the questions seem way too easy, it's because they are. Like, it'll ask you, what are the elements in water? And you're like, hydrogen and oxygen? Like, obviously. So it's meant to be easy because it's going to build as you go through. I do want to just draw maltose once with you so that you get the idea of how to do the maltose. On your drawing quiz, you're going to be asked to show the formation of these, which shows the two things separate. So in this case, the two glucose separate. And the formation would be here they are separate, and now here they are joined together as a maltose. What do we have to remove to join them? Got to get rid of a water, right? Okay, so here we are, we're going to draw, because it's maltose, I know it's glucose plus another glucose. So I'm just going to draw them twice. I'm going to do them a little bit smaller so I have enough room. Only use about half of your page for both of these pictures. So here we go. We're going to do carbon, oxygen, carbon. Actually, it's easier to lay out the carbons first and then do the bonds. You'll get better angles. Okay? CH2OH at the top. Down, up, down, down. For my hydroxyls. Everything else is a hydrogen. 1, 2, 3, 4, 5. So that's glucose. Do it again. I'm going to add it to another glucose. C, C, O, C, C, C. Now we're going to remove a water. We're just going to use the hydroxyls that are closest to each other in this diagram for this particular diagram, just because it's just easier for us to do it. We could technically use any hydroxyl and just move them around, but we'll make it a nice linear molecule here. So I'm going to take a hydrogen off of one of these, take an OH off the other, and then that leftover oxygen on the left will be paired up with the leftover carbon on the right. So then I just draw it out again, it becoming maltose. So I have my separate glucose. Notice that the ring structure never changed. So that stays the same. But I'm going to add in the other structure right away. The CH2OHs were not involved, so they're all the same. It's only the hydroxyl on the end of the first molecule and the beginning of the second molecule that are going to be modified. How are they modified? They get removed and left behind with just this oxygen that joins the two molecules together. So I can draw that next. There's the oxygen that now joins them. The rest of the molecule is identical. Down, up, down, and this is my next down. But this also starts as my first down of the other guy. So this would be my first down, and then up, down, down. Should there be a hydrogen on this carbon right here? Yes, absolutely. How many bonds does the carbon need? 4. It's missing something. It must be missing a hydrogen. And this again is one of the things that people miss when they're doing their tests. Both of these carbons need to have little hydrogens on them. Don't forget this pesky hydrogen, and then fill in the blanks with the other hydrogens. And now you've just drawn your first successful maltose. On the test, I'm not going to ask you for every version of disaccharide. I am specifically asking you to draw this one. When I ask for formation, I'm asking for the two molecules separated before and then showing me what they look like afterwards. There is one thing missing here. Can you guys guess what it is? Now you have the full formation. Because those hydrogens and oxygens went somewhere. They didn't just disappear off of those molecules. They became water, right? Sucrose, I'd like you to try it just so that you get the idea of how to join these. It'll be exactly the same thing as the picture you saw before, but the one you want to practice over and over again is drawing the maltose.